In this presentation, I'm going to show you some images of bacteria that were stained using the Gram stain, and I'll also explain how this process of the Gram stain works. This is showing you an image of Staphylococcus epidermidis. Under the microscope, this is using the oil immersion lens, so we're looking at this cell under 1000 total magnification. And as the name implies, these cells are cocci. They are circular cells, and you can kind of see the outline of those cells being circular right there. And you'll notice in this image that the cells appear purple. So the purple staining indicates that Staphylococcus epidermidis is a gram-positive bacterium. This next picture is showing you an example of a gram-negative bacterium. So what I did was perform the Gram stain on a sample of Escherichia coli, also known as E. coli. And you can tell from these images, this is at 400x and this is at 1000x using oil, that the cells definitely look pinkish reddish in color. Just so you know, E. coli is actually a rod-shaped bacterium. However, it's such a small, tiny rod that when you look at these under the microscope, you might be mistaken and think that they were cocci. So E. coli is a rod. Either way, it stains pinkish red, which makes it a gram-negative bacterium. Okay, so let me explain to you how this whole process of gram staining works. It's based on the principle that gram-positive and gram-negative bacteria differ in the composition of their cell wall. So if we take a look at this picture down here, this is kind of a slice through the outer layers of a gram-positive and a gram-negative bacterial cell. So here's the plasma membrane, so we'd have the interior of the cell down in here, and then this is the plasma membrane. Plasma membrane, remember, made up of those phospholipids in two layers, referred to as the phospholipid bilayer. So gram-negative and gram-positive bacteria both have a plasma membrane on the very interior layer of their cells. The next layer out is the cell wall, and this is where gram-negative and gram-positive bacteria differ. In a gram-negative bacterium, their cell wall has two major parts to it. It has a thin layer of peptidoglycan, and then it has on top of that another phospholipid bilayer that rests on top of the peptidoglycan. That's very different to the gram-positive cell wall, which is a thick layer of peptidoglycan. They do not have an additional plasma membrane that rests on the top. So when we do the Gram stain, the difference in the cell wall composition really makes a difference as to how those different stains are retained within the cell. So let me show you, kind of go through this process of the Gram stain and talk you through what's happening. So the first thing you do in the Gram stain is, or the first stain you add during the Gram stain is crystal violet. So crystal violet goes into the cell wall of both the gram-negative and the gram-positive cells. The next step in the Gram stain is to add the iodine. And what the iodine does is basically latch on to the crystal violet and allow it to all kind of stick inside the cell wall. The next step is to add ethanol or alcohol to the slide. And so what the addition of ethanol does is to remove this plasma membrane layer on the top of the peptidoglycan in only the gram-negative cell wall. So when you add the ethanol, it basically strips away or dissolves that lipid layer that sits on top. So this is what we have after we've added the ethanol. And because of the loss of that lipid layer in the gram-negative cell wall, the crystal violet and the iodine also disappear because they were stuck into that layer. When that lipid layer goes away, so does the crystal violet and the iodine stain. In the gram-positive cell, all of that stain stays in place because the peptidoglycan is unaffected by the addition of the ethanol. The very last step of the Gram stain excuse me, is to add the counterstain, or another stain called safranin, and that safranin is a red stain. So when we add safranin to the gram-negative cell wall, that stain comes in and attaches to the peptidoglycan, and when you look at those cells under the microscope, they appear pinkish red. When you add safranin to gram-positive cells, that safranin goes into the cell wall and stays in there. However, crystal violet, that purple stain, is much darker and overpowers the visibility of the red stain. So safranin's in there, but crystal violet basically masks that, and the gram-positive cells appear purple still when you look at them under the microscope. So hopefully that explains how we get those different cells, gram-positive and gram-negative, appearing either purple or pink under the microscope.